Good evening, Pratham. Okay. So, so today we'll be talking about the, you can say, spinal reflex, okay, and the reflex arc, the spinal reflex and the reflex arc. So I'm Rahul Saha, and last class we had talked about the entire circuitry regarding the action potential, resting brain potential, and also why does it propagate only in the forward direction. And also we had talked about, you can say, the specificities regarding the release of the neurotransmitters. Okay, so those things we had talked about. So today we'll be talking about the spinal cord, spinal reflex. So most of the reflexes are spinal reflexes. So reflexes are what? When we touch something, we just take our hand off, or it is just a type of, you can say, evolutionary mechanism which provides us protection, right? Which safeguards us from, you can say, heavy duty damage. That is the role of the reflex actions. Okay, so this is why reflex actions are extremely necessary. Now, when reflex actions are absent, what can happen is we can heavily damage ourselves. Okay, so sometimes the reflex actions only take the course of the spinal cord and not through the brain. The information does go to the brain, but after the effector muscle has caused a specific, you can say, action, or has perpetrated an action, after that it goes to the brain. Like, we feel that okay, we had touched something, but and that thing may be quite hot, or we might have got electrocuted, we have just experienced a current flow, but till that time we have already taken our hand off that place, right? That is there. So cranial reflexes are very few, spinal reflexes are more. So I'll show you just the diagram of a spinal cord first. Okay, spinal cord. I'll show you like this: dorsal horn anatomy, dorsal root. That will be much better diagram of spinal cord. Okay, this is the diagram that I wanted to show you. Okay, look at this diagram properly. I'll just open the site if it opens. This diagram is what we are talking about. Okay, this one. So this is the diagram that I want you to concentrate on. This kind of diagram is present in your NCRT and also in your reference books. Okay. But the thing is, the explanation is absent, but all these things are written. Okay. This diagram is present. The entire reflex arc is drawn. Okay. But the explanation is quite, you can say, uncertain, will only perplex you till a level, but the diagram is completely present. Okay, so the thing is, good evening, Samesh. So this is the diagram that I want you to see. Okay, this diagram. This is the diagram of the spinal cord, and this diagram is present in your books also. And this diagram, through this diagram, I can explain to you the entire reflex arc or reflex action. Okay, so in class 10th or lower classes, you might have learned that only spinal cord is responsible. That's all. But how and why, and what is the spinal cord made up of? Okay, so you can see in this diagram that the outer portion of the spinal cord, okay, the outer portion of the spinal cord is made up of white matter. White matter means the axons that are myelinated. Okay, they will be white matter. Just the opposite of the central nervous system. In the central nervous system, the cortical region, the top region, is the gray matter. The inner region is the white matter. In the spinal cord, it's just the opposite. The outer region, you can say, or the cortical region, is the white matter. The medullary region, okay, is the gray matter, as you can see. This is gray in color, okay, due to the presence of Nissel's granules. I told you it takes the, you can say, stain crescil violet and turns darker. So here we have the dendrites, we have the telodendria, we have the synaptic end bulbs, and we have also, we can have non-myelinated axons. So non-myelinated covering or unmyelinated covering is present here. So it looks like gray in color. That is gray matter which is present at the deeper region or the medulla of the spinal cord. Okay. Now you can see this thing. Okay. So in order to show you this, in order to explain to you this specific stuff, I'll draw my own version of spinal cord, okay, and explain to you what really happens. Okay, just hold on with me. I'll draw my own version of the spinal cord and explain to you what really happens. Okay, let's come to the whiteboard. Okay. So what happens is, if this is the spinal cord, okay, what you have is, here you have multiple rootlets which combine, okay, to form, this is known as dorsal root ganglion, okay, dorsal root ganglion. Now here also there will be multiple rootlets, they will all combine, okay, they will all combine and they will come out of the, you can say, interventricular foramina. A foramen is a specific hole, you can say, it's a specific orifice, okay, an opening through which the spinal nerves come out of the, you can say, backbone. So I'll just show you spinal nerves. That diagram will become clear for you. There are 31 pairs of spinal nerves, okay, this one I'm talking about. Yeah, you can see this diagram also. So this is a good image that I've got. So these are good images. You can see this, okay? So from here, here, these are the foramen, okay? So from here, the nerves, they come out, okay? The nerves, they come out. That is known as intervertebral, between the two discs, okay? If I show you also, I'll type it out and show you intervertebral foramen, okay? Foramina. This is, you can see, look at this image, okay, this is intervertebral foramen, the gap between the two vertebral discs through which the nerves can come out from both the directions, okay, it comes out from the both the directions. If you have seen, it is like this, okay, it is like this image, it comes out from both the direction, you can see, okay, both the direction it comes out. So every nerve has a pair, that's why. So you have five cervical nerves, you can say five cervical, okay, then, sorry, we'll be having 8 cervical nerve, 12 thoracic nerve, okay, then 5 lumbar nerves, okay, then 5 sacral nerves and 1 coccygeal nerve. These are all in pairs. So 8 plus 12, 20 plus 30, 31. So 31 pairs means 62 spinal nerves. So they come out like this, right? They come out like this through this portion. Okay, you can see this is the best diagram to specify the how the spinal nerves come. Okay, and one more thing I'll tell you is, just see here, you might have also heard, since you both are ISC students, you might have also heard that spinal nerves are known as mixed nerves, right? Might have heard this. Spinal nerves are mixed nerves. In class 10 you might have heard. Now why are they mixed nerves? That also your teacher might have given you an explanation, a layman explanation, that it carries both sensory and motor neurons. Okay, carries both sensory and motor neurons. Now I'll explain to you how that happens. If you come here to this image, okay, just look at this image. Let's go one step back. And this I found a very good image here. This one. Just look at this image. Yeah, just zoom it for you. Okay. Look at this image. This is what I was drawing here. Okay, this is what I was drawing here. The rootlets have not been drawn here. Directly this, then this, you can say bulb type thing. Okay, now come here to this diagram. Okay, now the thing is, this is the dorsal part of the spinal cord. Dorsal means posterior. If we cut this is my spinal cord, okay, this one, if I cut it like this, that is the transverse section. So part facing this one is the anterior part, part facing the back after getting cut transversely will be the dorsal or posterior, anterior or ventral. Okay, ventral means anterior front, dorsal means posterior back. Anterior means in the front, dorsal means at the back. Technically, they are known as ventral dorsal, anterior posterior, or complete layman front back. Okay, so now there are multiple rootlets, means there are multiple, you can say, axon fibers which are emanating, okay, which are coming out from the, you can say, dorsal part of the spinal cord. Okay, now this is known as the dorsal root ganglion. Why ganglion? What is ganglion? Ganglion, I told you, is the cluster of cell body in the peripheral nervous system, right? Now, central nervous system is what? It is a culmination of brain and spinal cord. Now you have come out, you have left the spinal cord, means now here all this part is in the peripheral nervous system. So these nerves, okay, the cell bodies of these nerves are present here. Okay, they are known as dorsal root ganglion, and then they combine here. Okay, and then they come out of the, you can say, the intervertebral foramen. And these nerves, these dorsal root ganglion nerves, this dorsal root ganglion contains, very very important, this diagram is given in your book also. Okay, this contains the sensory nerve. Okay, meaning the afferent fibers. Afferent means coming towards. So whatever you feel, touching this, I'm feeling this, so it is coming, that those nerves are coming and they are reaching my spinal cord through the dorsal root ganglion, and this is the cell body cluster of the sensory nerves present just close to the spinal cord before it has exited through the intervertebral foramen, but it has exited the spinal cord. Okay, so this contains the sensory nerve cell bodies. Okay, cell bodies, that is, you can say, the afferent nerves, means the signal is going towards the spinal cord, means towards the brain. And the ventral nerves, they carry the motor neurons. They carry the motor neurons, but their cell body is not present in the root, means outside the spinal cord before the, you can say, intervertebral foramen. Their cell body is present inside the spinal cord, you can say, in the gray matter present inside the spinal cord. So their cell body is not outside the spinal cord, their cell body is present in the gray matter of the spinal cord, that is known as nuclei, not ganglion. Okay, so they do not have any bulgy part here, just like this. They do not have, you can see the ventral one doesn't have any bulgy part. These are the motor neurons. Okay, these are the motor neurons, and they mix up here together. Okay, they mix together, they combine together and come out. Okay, from the, you can say, intervertebral foramina at this point. Okay, they come out, and that is why they are known as mixed nerve, because see, this is sensory, this is motor, so this is a mixed nerve, the spinal nerve. That's why technically, conceptually, it is known as a mixed nerve, you can say. Okay, so is it clear till now? No issues, Gayatri? Is it clear till now? Or Gayatri, shall I repeat it for you? Till which part did you get? Till which part did you get, Gayatri? Like, was it clear anything, or shall I explain again? I was explaining the entire spinal cord here in order to explain the specific reflex arc. Okay, this diagram you might have seen in your book also given of reflex arc. So, okay, I'll explain it again. Okay, so just listen from this diagram. Okay, so this is the diagram of the spinal cord. Okay, so this diagram is given in your NCRT book also. You must have seen. Okay, this flat like diagram. So this is the diagram of the spinal cord. Okay, now in a spinal cord, the outer matter is the white matter. Okay, cortical matter, inner is the gray matter. So you can see this color. This is due to the nasal body. They are taking the crescent violet stain and they are becoming dark, right? This concept you know already. That is the gray matter. This is the white matter, the cortical region. Now, in the brain, we know that the cortical region is the gray matter, the internal medullary region is the white matter. In the spinal cord, it is just the opposite. Okay, just the opposite. Now, you also may have heard that spinal nerves are mixed nerves. Okay, how are they mixed nerves? You can see these nerves coming out. Okay, from the spinal cord. This is the gray matter. This is the white matter. Now I'll explain to you how does it happen. Now come to this diagram. Okay. Before that, come to intervertebral foramen. Okay. So this is the intervertebral foramen. You can see means foramen means an orifice or hole or opening through which the spinal nerve can come out. Okay. So spinal nerves, they come out in pairs like this. You might have seen the diagram also. It comes out in pairs. So this is the intervertebral foramen. This is one more diagram. So it comes out in pairs like this. So there are 31 pairs of spinal nerves. Okay. So 8 sacral, sorry, 8 cervical, 12 thoracic, then 5 lumbar, then you can say 5 sacral and 1 coccygeal. Total 31 pairs means 62 spinal nerves. From each point, they'll diverge into two sides like that. Now, before diverging, before diverging, this is what happens. This is the intervertebral foramen, the space between the two discs. Now, this is what will happen. Now, here this region, okay, is the intervertebral foramen you can think of. Okay. Now, this is known as dorsal part, this is known as ventral part. Don't worry about the root here. Okay. Don't worry about the root. So, first of all, concentrate on dorsal and ventral. Okay. What is dorsal? What is, you can say, ventral. Now, we all know that dorsal means backward, ventral means front. Okay. Dorsal root and ventral root you can think of. Okay. So, what I want you to explain, to know is to understand and comprehend this. Suppose this is my spinal cord. Okay, this is my spinal cord. I cut myself like this straight. Okay, that is a transverse section. This is a spinal cord. You cut it like this. This is the transverse section. So this part is facing my chest. This part is facing my back. So this part will be known as the anterior part or the ventral region. This part, the backward part, will be known as the posterior region or the, you can say, dorsal region. Okay, so that is known as the dorsal root. The dorsal root is one of the two roots which emerge from the, you can say, spinal cord containing multiple spinal nerves, because what you have is many rootlets. So this is rootlet, one, two, three, four, five, many rootlets, then this will become a nerve. Many rootlets, this will become a nerve, and they will just fluff up like this one. This is fluffed up region. This region is known as dorsal root ganglion. Okay, that is known as dorsal root ganglion. Now, what is a ganglion? A ganglion is a cluster of cell body in the peripheral nervous system, right? Cluster of cell body in the peripheral nervous system means central nervous system is a combination of brain and spinal cord, and this nerve has already exited this spinal cord, means it has exited this central nervous system. Now it has reached from this point onwards, this is the peripheral nervous system. So the cell bodies are clustered in the peripheral nervous system, therefore it is known as, you can say, ganglion, and it is a part of the dorsal root. Okay, so it is known as dorsal root ganglion. Too many cell bodies, so it is a thickened portion. Okay, now this specific nerve is of which type? This specific nerve is of sensory nerve type, means the afferent nerves. They are all, you can say, the afferent nerves. I have already labeled, they are the afferent nerves, okay, which bring the information in this direction to the spinal cord. This is the spinal cord and the brain. Okay, so these are all sensory neurons. Their cell body is present outside the nerve, outside, sorry, the spinal cord, but this is the intervertebral foramen. Before the, like, after crossing the intervertebral foramen, it is present. Okay, in case of ventral nerve, the cell body cluster is not present outside the spinal cord, thus it is not a ganglion. It is present somewhere here only, inside the inside the spinal cord. So it is known as a nucleus. Okay, and ventral part takes the information away from the spinal cord. That is, it is an efferent neuron, or made up of many efferent neuron or axons known as fibers, also known as motor neurons. Okay. So now look at this diagram. This is sensory bringing information to the brain. This is motor neuron taking information away. This is the intervertebral, you can say, foramina or foramen. Here both the nerves are getting mixed or not. Both the nerves are in contact. Therefore, the spinal nerve is also known as a mixed nerve because it is carrying both sensory and motor nerve. That is the reason. That is why spinal nerves are known as mixed nerves. And after coming out of the intervertebral foramina, they divide into two parts. Okay, you can see one of the sensory goes with one of the, you can say, motor. Here one sensory goes with the motor, and then the two mix. Just see, try to understand here. One blue and one, you can say, red is here, one blue, one red, but after crossing this specific intervertebral foramen or intersection, both the blues are combining together, both the reds are combining together. Okay, so this is known as ventral ramus, this is known as dorsal ramus. Okay, this sensory and motor neuron, okay, goes to the back side of our body. All the back region of our body, it carries the sensory and motor function. It provides to the back region. Ventral provides to the front, our hands, our front legs, this chest region. Okay, that is all the sensations that we feel, sensory motor that we can do from the front part is considered or is conducted by the, or perpetuated by the nerves coming through the ventral ramus. Okay, so is it clear now till this part? Till this part, is it clear? Gayatri, Pratham, is it clear? Samesht, till this part? Or do you have, if you have any doubts, just ask me. This is a very conceptual thing, this diagram. And if the concept is clear, you will become the master of neuroscience for the rest of your life. Okay, because this diagram is drawn in the NCERT book. Okay, but explanation is given very awkward explanation. Okay, like very, you can say, indecipherable explanation is given, you will never be able to understand the concept of reflex action. Okay, so is it clear till now, till this part? Is there any query till this part? So this is how the spinal nerves, they make the connections with the entire body. Okay, this is how the nerves emanate from the entire body. Okay, Gayatri, good. So next is simple reflex action. Okay, in all the simplicity, reflex action is what? Okay, reflex action is the nerve coming from the, you can say, receptor. Receptor means, suppose this is my hand. Okay, this is my hand. If I get the diagram of the reflex action, okay, much better. Okay, reflex arc diagram. Okay, we got a good, very good diagram here. This is also a good diagram. Okay, you can see this diagram. So this is one of the best diagrams of reflex action. Okay. Now what happens is, suppose this is the muscle of your, you can say, or this is something as skin. Okay. You pricked a nail through your skin. Okay. Inadvertently, you didn't know. You just pricked it, and just like that, you will take your hand off the prick, right? The nail. Sorry. You will take your hand off the nail. You pricked it, and what will happen is, what has happened? This is the effect, this is the receptor. So there are receptors present here, and then the signal is going, okay, this is the ganglia, okay, to the spinal cord. They have synapsed with an interneuron. Interneuron has synapsed with the motor neuron. Motor neuron is going and telling, is conveying the, you can say, information to the muscle. Means this muscle, this one, the brachialis muscle, this muscle will be present. Okay, it will take your hand off, okay, from that place. Okay, so this is our radius ulna, so brachioradialis, you can say. So this will just take your hand off. Okay, so that is your reflex action, quick reflex action. Okay, one more reflex action is known as patellar action. So patella means the kneecap. So I'll show you that diagram also. Just look here. Okay. See, knee jerk reflex it is known as. Many times it is asked also. Okay. If I can zoom in the image. Okay. I've zoomed into the image. Just look at this image. Now what happens is, when you give a visit to the doctor, okay, the, suppose you are not able to feel your knees properly, or feeling numbness in your knees, so the doctor will use a hammer and hit on the knee and check your reflex action. If the time spent is, you can say, admirable, or it is too much, too, or you can, too much means it is delayed, or it is just adequate. Okay, so that will be checked, and based on that, your nerve impulses, the nerve conduction velocity can also be estimated. Okay, if it is too slow, or if it is adequate completely. Okay, so then what you can do is, if you just do the hammering on the knee like this, so what do you expect? What you will do is, suppose this is the knee, I am putting the hammer here, my knee will cock back, right? Cock in front, sorry, to take away the jerk, or to just evade. That is the reflex, right? If you hit the knee, This is the knee. The knee will go in the forward direction, right? The knee will go in the forward direction like this. Okay, you can see the direction also here. It will go in the forward direction. So that is the reflex of the hammer. So what happens is this is a type of monosynaptic reflex arc. This one was polysynaptic. Okay, this one which I had shown you was polysynaptic. You can say one synapse, one interneuron, another synapse. So more than one synapse, polysynaptic. But this one, if you see, directly the sensory neuron is coming and synapsing with the motor neuron. There is no interneuron here. Directly sensory neuron to motor neuron synapsing and then going into the, you can say, directly into the stretch receptors, okay, of the quadriceps, okay, of the quadriceps, and that quadriceps muscle is contracting and then we are able to throw the knee forward. Okay, so that is what is happening. So this is a monosynaptic, you can say, a monosynaptic reflex action. That is known as the patellar reflex action. It is different. You can have polysynaptic also. The polysynaptic reflex action will have nothing but it will have the participation of an interneuron. So in competitive exam, just write participation of an interneuron. That's all. Okay. Now, this is monosynaptic one. Okay. But the thing is, have you ever thought of how does your muscle get the signal? Okay, how does your muscle get the signal? So I'll just try to explain to you in a very simple term. You have already learned the chapter muscle. So the muscles can be divided into two parts or two types based on their position. Okay, the skeletal muscle I'm talking about, not any other muscle, not the types of muscle. The skeletal muscle can be divided into two parts based on their position in any organ. Okay, suppose this is my limb. Okay, so suppose this is the connective tissue, yeah, on my limb. Okay, and these are the muscles which are originating from the tendon. Okay, muscles that usually originate from the tendon. Okay, these are the muscle fibers which are originating from the tendon, and this muscle fiber is originating and is connected to the connective tissue. It is not originating from the tendon. Okay, it is just originating from the connective tissue. Okay, so it could be like this or it could be like this. You know, muscle cell. Okay, huge muscle cell. Okay, muscle fiber or muscle cell. Now these muscle cells, okay, these muscle fibers which are originating from the tendon and are surrounding the connective tissue capsule. By the red what I have drawn is a connective tissue capsule. Okay, this is a connective tissue capsule. So I'll name it also. Connective tissue capsule. Okay, so these muscles are known as extrafusal muscles. Okay, these are known as intrafusal muscles. Okay, so this is only for your competitive examination. This tip for you. Okay, this won't be asked in any other exam except for NEET. Okay, so this specific thing, extrafusal and intrafusal, I'm just giving the tip for you in order for you to be completely thorough with the also the NEET part. Okay, the reflex action part is done. This is the NEET part that I am explaining to you. So this intrafusal muscle, extrafusal muscle, they are one thing, it will improve your understanding also. So these muscles are the high tension muscles, means they generate a lot of tension via, you can say, sliding filament theory. They do not generate so much tension. And the sensory neurons, okay, the sensory neurons, they wrap around, you can say, intrafusal muscle. They don't wrap around the extrafusal muscle. That is why I'm including this in the, you can say, reflex action. Okay. So what happens is, when you are hit with something, okay, or something, suppose you are here, a 40-pound thing is being dropped on your hand, so this entire thing is getting stretched, okay. So the intrafusal muscles, okay, here what you have, something known as the spindle fibers, okay. The spindle fibers are the stretch receptors, okay, like proprioceptors, they are the stretch receptors. They will know that stretch is there. All of a sudden a huge drop of, you can say, a weight or mass has occurred, that stretch has happened, and now what they'll do is they will try to protect your entire arm from breakage or from any further injury or damage. So now this sensory muscle fiber will go down this route, okay, and the motor nerve will come and will wrap around the extrafusal. Motor nerve will wrap around the, you can say, extrafusal also, and some motor nerve around the intrafusal also, and then they'll make it contract, and this will not contract that much, it will contract little, but it will provide the sensitivity, you can say, paradigm to the extrafusal muscle, that this much contraction is enough, okay, only this much contraction is enough, okay. So that is the main difference between intrafusal and extrafusal. But in higher classes, when you become a doctor, then that time you will be studying about, you can say, alpha sensory neurons, okay, alpha motor neurons, okay, those things we will be studying about, like the gamma motor neurons, okay, they innervate the, you can say, intramuscular, sorry, intrafusal muscle. So those things are not needed right now, but later on we will be learning those also. The thing is, first the stretch receptors, okay, the something fell on you or something you got pulled, is present on the intrafusal muscle, means the muscles which are present inside the connective tissue. That was my main aim, why I described to you this, so that your entire reflex concept is clear, okay. And then the sensory receptors are present, then it is conveyed through the, or via the sensory nerve to the, you can say, spinal cord, and the motor nerves then come to the extrafacial, sorry, extrafusal and intrafusal, and make the extrafusal contract more, so that it is kind of a, you can say, compensatory effect or compensatory contraction to save you, to save your specific organ, or that organ which was under a specific attack or some accident occurred supposed to that specific organ. So that is known as your entirety of the, you can say, this entire extrafusal intrafusal muscular dynamics behind the reflex arc, okay. So this is how reflex arc happens, okay. So after that we can learn little bit about the anatomy of the spinal cord also. Very basic anatomy is asked in class 11th, not that deep anatomy, okay. Simplest anatomy is, this is the gray matter, this is the white matter, number one. Dorsal root, ventral root we know. Then dorsal root ganglion outside, ventral root ganglia, sorry, ventral nuclei will be inside. Okay, that is fine. Now, here the white part, okay, and the gray part. The ventral, okay, white part towards the ventral side, okay, that is known as the ventral horn. This and this part, okay, this and this part will be known as ventral horn, means the gray matter towards the ventral root or towards the forward side, means this is the forward side, so the gray matter towards the forward side are known as horn, Okay, gray matters are referred to as HORN, horn in case of, you can say, gray matter, okay, are referred to as horn in case of spinal cord. You can write it down, very, very essential anatomical term. White matter, okay, was earlier referred to as funiculus, okay, but now it is referred to as column. Okay, now it is referred to as a column in case of, you can say, spinal cord. So what happens is we have ventral horn, dorsal horn, and lateral horn. Okay, very, very easy. You can see in this diagram. Okay, this one I think, yeah. Ventral horn here, lateral here towards dorsal side, dorsal horn here. Same way, dorsal column here and here. You can see between the lateral horn and ventral horn, you have the lateral column, and towards the ventral horn, you have the ventral column. Column means white matter. So this is the entire specificity of the, you can say, spinal, you can say, cord, right? Spinal cord related anatomical details. So that is all that you need to know about the spinal reflex, right? About the spinal reflex. So now we can talk about something known as the next topic, that is known as neurotransmitters. Okay, we can talk about the neurotransmitters. Now, what are neurotransmitters? Neurotransmitters are the molecules which we talked about, which are secreted, right, from the synaptic end bulb at the synaptic cleft or synapse. Okay, so there are two things I'll tell you. One is neurotransmitter, another is, you can say, neuromodulator. Okay, one is neurotransmitter. Another thing is also asked, known as neuromodulator. So just by the name you can say what is modulator. So neurotransmitter, if it is released, it will directly bound to the postsynaptic neurons, dendritic spine, means the receptors present on the dendritic spine on the postsynaptic neuron. It will bind to it and it will cause effect. It will cause depolarization, it will cause hyperpolarization, okay? Then it can cause action potential to begin. Like it will bind to the ligand gated acetylcholine channels, which I had explained to you in the last class, and then it will cause the action potential to begin or action potential to dampen. Both it can do. But neuromodulators, they do not bind to the dendritic spine receptors. What they, and even if they bind, okay, they do not cause any depolarization or hyperpolarization effect. Okay, that is the main thing. They do not cause either depolarization, like neurotransmitters bind to the ligand gated channels and cause sodium influx depolarization. Okay, they do not cause depolarization and repolarization, sorry, and hyperpolarization. Hyperpolarization, what they do is they can modulate, that is modulator, they can modulate or change the effect of the neurotransmitter, the effect of neurotransmitters. They can change the concentration of neurotransmitter release also. Okay, so these are very, very special molecules known as neuromodulators. Only this much you should know, that it will cover all kinds of entrance tests regarding the neuromodulators. Okay, one more thing I'll tell you, is something known as synaptic delay. Okay. So directly synaptic delay is asked in competitive examination. Synaptic delay is of 0.2 to 0.5 millisecond. Okay, you can note it down. 0.2 to 0.5, you can say, millisecond. So synaptic delay occurs. What is synaptic delay? It is a delay that occurs between the arrival of the action potential at the axon terminal or synaptic end bulb or telodendria or varicosity and the effect on the postsynaptic membrane. Okay, means the action potential has arrived at the axon terminal. Now depolarization will be happening, calcium ions will enter the membrane, the vesicles will contain neurotransmitters, they will fuse with the presynaptic neuron axon terminal plasma membrane, and then they'll be released, then they'll bind to the postsynaptic neurons receptor. Okay, then they'll bind to the receptor of the postsynaptic neuron, means here they'll be binding with this, and there they'll be binding with the receptor of the postsynaptic neuron, and then depolarization will happen. So till the action potential comes, so when the action potential comes, from that point till the depolarization or hyperpolarization happens, that is the time it is known as synaptic, or that time is referred to as synaptic delay. So synaptic delay is not caused generally by, you will be thinking that the neurotransmitter release, then neurotransmitter binding takes a lot of time. No, because the synaptic, you can say, cleft is very small, 20 nanometer only. Okay, and diffusion rate is very high. So some nanoseconds, or you can say microseconds, are wasted there. Maximum time from the advent of action potential at the axon terminal, okay, to the effect on the postsynaptic neuron goes in the, you can say, opening of the calcium ion channels. Okay, so most of that delay reflects the time involved in calcium ion influx and neurotransmitter release, not in the neurotransmitter diffusion, because the synaptic cleft is very narrow and neurotransmitters can diffuse in a very little time at very high speed. Okay, so calcium ion influx, okay, then the entire intracellular signaling pattern which will lead to the formation of the vesicle. Formation of the vesicle, vesicles will contain thousands of molecules of neurotransmitters, suppose acetylcholine, and then that fusion of the neurotransmitter vesicle with the presynaptic axon terminal plasma membrane, that takes the maximum time. After that, when the neurotransmitter is released, then subsequently in microseconds it can be, or it can reach the postsynaptic, you can say, dendritic receptors of the neurotransmitter. Okay, so you can say although a delay of 0.5 millisecond is not very long, but in that time, if you check the speed of type A fibers, the action potential can travel more than 7 centimeter or 3 inches along a myelinated axon. Okay, so that is there. So the delay is delay. Okay, you cannot just say it is not a delay. Okay, so now what we have is, we have two classes of neurotransmitter. One is excitatory neurotransmitter, one is inhibitory neurotransmitter. Okay, now excitatory will do what? Excitatory neurotransmitter will do what all steps? So excitatory neurotransmitters like glutamate, okay, glutamate, amino acid, then aspartate, these are all excitatory neurotransmitters. What they do is, they cause depolarization. We have already seen in the graph, okay, we have already seen in the graph that if depolarization happens, that will only lead to the perpetuation of action potential. Otherwise action potential cannot travel, because it is minus 70 millivolt. Okay, this is the time x-axis, y-axis. Okay, now if the graph doesn't go in this direction, okay, will action potential travel in the forward direction? It can never happen, okay, because once you need to reach minus 55 millivolt, then voltage gated, you can say, sodium ion channels will open up, okay, then they'll open, and then it is known as plus 30, okay, then here it will close, and then entire thing that I had explained you will continue, and action potential will propagate further. Refractory period, then Na plus K plus ATPS will revert the concentration after, you can say, 100 action potential channels, when the concentration of the ions will start to go up or down, okay. So the thing is, without this thing, means without depolarization, this is the graded potential which is reached after you have ligand gated sodium ion channels activated by the binding of acetylcholine or glutamate or aspartate, right? This is repolarization. So this is all influx of Na plus, this is K plus efflux. Now tell me, without Na plus influx, can current propagate forward? It can never propagate forward, right? So any excitatory neurotransmitter means what? It will cause the flow of current to be maintained. It will not cease, it will not stop the flow of current from presynaptic to postsynaptic neuron. Means the neurotransmitter, when it is released from the presynaptic neuron, and it then binds to the postsynaptic neuron, it will then perpetuate the flow of current, okay. It will not slow down the current flow or not cease or stop current flow. That is the meaning of excitatory neurotransmitters. But if the neurotransmitter like gamma aminobutyric acid, which causes sleep, okay, that can do what? That can cause hyperpolarization like this. It is already at minus 70, okay, millivolt. This is time, x-axis, y-axis, okay. So instead of opening calcium ion channels, okay, it can open the chlorine ion channels, okay, and let the chlorine enter. So when the chlorine will enter, it will become more negative or not? So the voltage will drop. It may drop till minus 90. So will action potential ever propagate? No. So one of the two things, the, you can say, inhibitory neurotransmitters can do. Either they can cause K plus efflux. By causing that also, they will drop the voltage to more negativity. The voltage will become more negative. Or they can cause chlorine ion influx, because you are bringing negative charge, so the voltage will again become more negative. Once it has become more negative, instead of reaching minus 55, more negative it is reaching, you can say, plus, sorry, it is reaching minus 90. So there is no way that current can propagate. The current flow will cease there, and then we doze off to sleep. So that is one of the functions of, you can say, GABA. That is an inhibitory neurotransmitter. Inhibitory neurotransmitter. It binds to the GABA A receptors. Okay, mostly GABA A are there. Receptors on the postsynaptic neuron. Okay, inhibitory neurotransmitter. Transmitter. This causes us to fall asleep, sleepy. Okay, and all the most of the, you can say, tranquilizers or sleep medications are derivatives of GABA. Means they mimic the structure of GABA and bind to the neuron to cause the same effect and to cause sleep. Okay, they are derivatives. They are derivatives of GABA. Okay, to you can say alleviate something known as insomnia. Okay, like alprazolam. That's the compound, okay, pharmaceutical compound which is used by the patients to alleviate their insomniac or their insomnia disease. Okay, so that is the work of GABA. It causes hyperpolarization. Okay, hyperpolarization, and thus is an inhibitory neurotransmitter. Okay, so I hope this lecture is clear to you. If you have any doubts, you can contact me on my personal number and I'll be replying as soon as possible. So do you have any doubts today? Any doubts regarding the excitatory inhibitory neurotransmitter or the reflex action, the reflex arc and spinal cord anatomy? Any queries, any clarifications? Is everything clear or any kind of doubts are remaining? Okay, good, Gayatri. Okay, then so let's meet in the next doubt class. Okay, then. Thanks a lot. Okay, good, Pratham. Let's meet in the next class.